starting to affect not only the mutations on the proteins that affect the cell cycle, but also the proteins in the surface coats, and now they don't hold on to each other as well, and the cell starts to break down, and the tumor mass that is growing out of control starts to invade neighboring tissues, and potentially starts to lose its adhesion. And these cancerous cells look really wacky. They don't look like normal cells. They're all spiky looking. They have really weird surface proteins. They're letting all sorts of stuff back and forth through the membrane that they shouldn't. All the proteins are breaking down and things like this. And once those proteins that are holding that mass and those contiguous cells together break down, they will start breaking off. And if they get into the bloodstream or the lymphatic system, they can then start moving around the body. And if they then clog in another part of the body, that tumor can start developing in other tissues. And that is when the tumor is said to have metastasized. Okay? Now, what causes those genes that are controlling the cell cycle to suddenly go out of whack? Well, one of the problems is that A, there's a lot of genes that control the cell cycle, and B, there's a lot of different ways that they can go out of whack. So that's why it's hard to potentially pinpoint what causes cancer. Right? One of the things that can happen is if you have a sequence in the DNA where the genes are being read, you can have sections of DNA when the cells are dividing getting transposed or moved around. Right? And if they're transposed and the section in front of them, which is where typically the DNA polymerases or transcription factors bind, cause the gene to be read, if that just happens to be put into an area where it's really hyperactive, where that it's being shoved into an area where there's a gene that normally is read for everyday function, and this one's not supposed to be read quite so frequently, suddenly you start making an abnormal amount of your growth-stimulating factors, proteins. You also can get copies of genes. Cells regularly copy out and sometimes amplify or make sort of duplicate copies of particular genes. This is thought to be really important in evolution because one of the things that's a big problem in evolution is if you've got a gene that's really functional, right, you can't tinker around with it because you need what that process product is that gene makes. But if you make a duplicate copy of it, this one can go along making all your proteins, and you can tinker around and let mutations arise in the new copy of it without breaking up the old function. And so gene amplification occurs in the chromatin to quite a bit of extent. But if it happens to occur in the wrong thing, then suddenly you can start to produce a growth-stimulating hormone that is actually in excess. Right? Or you can do things like you can have a mutation in the controlling region upstream that controls whether the gene is being read or not. This is where the transcription factor would bind, right? And controls that the gene is now read. If that's mutated and it just basically it doesn't wait for a signal, it just reads the gene whenever it wants to, right? Then you're going to get more of this stuff growing. Or you could actually have mutation in the gene itself that makes the protein hyperactive, right? So there's a whole pile of different ways that you could go from a gene that is normally producing, say, one of these little balls to ones that is producing way too many of these proteins, or a protein that is hyperactive and it just basically keeps going and doesn't slow down or stop. These things are uncomfortable. I don't know how you did it. Now, one of the other things that can happen is if you break up proteins that are part of the regular cell cycle that are part of this transduction pathway. If you look at, we have our tyrosine kinase here that has just received a growth factor. One of the pathways that this will typically enhance in this particular gene is it will activate a G protein on the surface called RAS. And RAS, once activated, starts a phosphorylation cascade. Right? That then does a final activation of this transcription factor. Transcription factor binds onto the gene and starts reading the gene, producing a protein that stimulates or kickstarts the cell cycle. This could be something like cyclin, right? Now, this is the normal factor. This will not get stimulated unless the cell receives growth hormone. All right, so it waits for that signal and goes through this pathway. However, one of the mutations that you can actually get that becomes a cancerous, and this is related to colon cancer, is that particular gene, G protein, this RAS protein, can accumulate a mutation. And if that mutation is one of these ones we call a proto-oncogene, if that mutation arises, and this is heritable, so you've got a predisposition for this particular kind of mutation, it will kickstart the phosphorylation cascade without actually receiving the growth factor. And this will basically abnormally stimulate the cell cycle division. Now, we normally have process checks and balances. One of the things to remember is we have a cross-check where these two pathways can actually interchange and crosstalk to each other. So if it suddenly goes, well, what the hell is going on? There's this big accumulation. We haven't received this thing. Another pathway can come in and cut off this or produce another gene that actually suppresses that product. All right? And what becomes problematic is if the cell starts dividing too quickly, right, it'll start dividing out of phase, won't have time to cross-check all its DNA duplication, and you can have a mutation in another gene. And you could have a mutation that occurs in another part of the pathway. Now, this is a slightly different one. This is from a melanoma. One of the things that can happen is if you have damage in your DNA, and this can happen from things like while you're copying your DNA, sunlight can actually break the strands, and you want the cell to say, whoop, stop, I need to fix this before I go into division. Right? And one of the pathways that you would get that, sort of say, hold on, put the brakes on, I need to stop, I need to cross-check everything, is if you get damage in the genome, it will activate another protein cascade, activate a molecule, or an active molecule called P53, which binds to the DNA and actually transcribes a protein that stops the cell cycle from advancing. It basically is like a pause button. All right? Holds the cell there until it fixes everything and then goes forward. And this is an example of how you can get crosstalk. But if a mutation now arises because you're dividing too quickly in P53, then when it gets hit by this phosphorylation cascade or protein kinase cascade, it can't do anything. So it can get the signal, but it stops the signal at this point. So nothing then gets made to slow the cell cycle down. And the result is you can get a stimulation that has a lot of overstimulation without anything that can block it or pause it, and the cell will start to divide. And it will divide too quickly, and then the next time it'll have a few more mutations, and then a few more mutations, and then a few more mutations, and it will start accumulating these until it becomes a full-blown cancer cell. And this is one of the things that happens with colon cancer. This is also close to my heart because my mother is a survivor of colon cancer. Because this is heritable, I have to go through all this kind of screening for colon cancer because I have an increased susceptibility of one of my genes becoming an oncogene. Right? Now, what would then happen is if you lose the control of a tumor suppressor gene, like APC, you would start getting benign lumps or polyps, and this is how typically people are screened for this. Right? This polyp starts growing actively, and then if that basically activates the RAS oncogene, then you start getting abnormal growth. Sometimes that leads to the loss of a tumor suppressor gene, DCC, and you get a larger benign growth. And then if that finally leads to a tumor suppressor gene, P53, being lost, then that can basically cause additional mutations, and now you've got a malignant tumor. Right? And this is basically how the cancer goes. Yep?
So, if you have a benign tumor, the chances are pretty likely that it's going to get malignant, or can you just leave a benign tumor?
Benign tumors, typically what will happen is if you have a benign tumor, if you're going in for screening, if you, like the screening for this, you have to go in for colonoscopies. Thank God they knock you out for this, because they stick an orthoscope up your backside and they look for these polyps, right? And if they find them, then they cut them off so that the chances are reduced for these. If you're going in for things like breast screening, if they see a lump, then they basically get you going back a lot more frequently to determine if that lump is changing shape or anything like that. As soon as you start to see a change in that shape, that's when you're looking at things like lumpectomies. Get it out of that benign stage before it can actually start to get into a malignant stage.
And there's no overlap? It can't be benign, but let go of some cells? It's entirely either benign.
Yeah, it's either benign. As soon as it starts letting go of anything, then it's said to be malignant. And that is why it's really important for the screening. And this is how they do a lot of preventative is pre-screening. The more you screen for these things, the more you can catch them at a benign stage, and as a result, you can potentially get them out intact before they actually metastasize. Right? And this is why you've got the importance of essentially sort of vigilance and screening.
But if you have one benign, doesn't that increase your chances of having other benign ones?
Not necessarily. It depends on what caused that benign one in the first place. Right? If it's something like a melanoma, it could be just happened to get hit by a particular carcinogen, like a UV light or something like that. Removal of it doesn't necessarily mean you're going to get another one, right? But if it's something that's heritable, right, then what you're basically doing is you're sort of saying, okay, I have a greater risk of actually having a proto-oncogene become an oncogene, and so you do more screening, and that's the kind of preventative. But you do have an increased risk factor. It doesn't mean you will get it, because all sorts of environmental things, kind of your diet, things like that, how much stress you have, or things like this, will also affect the chances of that proto-oncogene becoming an oncogene. It's not a one-for-one, right? It's just you have an increased probability over the general population. All right? And I believe that was my last slide. So we have a little bit of time. I would like to continue on, but this does examine